that focuses on agricultural and environmental issues in the developing world. CABI also has a strong interest in the creation, curation, and dissemination of scientific knowledge. Ivan is based at the CABI Center in Nairobi, Kenya. His work involves the development and implementation of invasive species management initiatives using integrated pest management approaches, and works with national, regional, and international stakeholders. He's an expert in classical biological control. Tilly Collins is on the Faculty of Natural Sciences and is a member of the Center for Environmental Policy at Imperial College, London. She's a trained ecologist and has developed scientific interests and expertise beyond her community ecology roots. She's an entomologist, an arborist, an urban ecologist, to name a few of the multiple hats she can wear. Tilly is also actively involved in science outreach. Perhaps most relevant to today's webinar is her significant experience thinking about and working on risk assessment and management challenges associated with genetically modified mosquitoes that she gained in part while working with the not-for-profit scientific consortium Target Malaria, which is developing Anopheles gambiae mosquito population suppression technologies based on Gene Drive for the purpose of reducing the burden of malaria in Africa. I'm delighted that both of you are joining us today. Ivan, you're going to be a first speaker, and I'm ready to hand this over to you. Let me know when you have your presentation up on the screen and ready, and I'll confirm.
Thank you very much. Hello, everyone. My name is Rwomushana Ivan, as Dave has introduced, and I'm going to be giving a talk about the risk versus benefits, trying to understand the risks versus benefits of classical biocontrol for crop pests, and I'll be looking at case studies from Africa to open our minds to what the kind of work we've been doing when it comes to classical biocontrol in this region. But before I start my talk, I would like to just quickly give a brief overview of CABI, given that we have a global audience and many of you may not know about CABI. We are an international not-for-profit organization, which is intergovernmental, and the work that we do seeks to improve people's lives by providing information and applying our scientific expertise to solve problems that affect agriculture and the environment. CABI was established by a UN treaty, level treaty agreement, as early as 1910. We are more than 100 years old. It is owned by 15 member countries, and all these member countries have an equal role in what we do in terms of our governance, our policies, and the strategic direction that we employ. We address issues of global concern, such as food security, food safety, and we do this through research and international development cooperation. So we cooperate with a number of agencies globally to undertake the work that we do. And of course, our classical biocontrol work spans more than 100 years, several decades, 100 years in terms of what we've been doing in this area of biocontrol for management of invasive pests that affect our crops. CABI is quite widespread in nearly all the continents. So what you see on the slide right there is where we are located. Our team is roughly 450 staff, and we are spread around 26 locations. And CABI Kenya is where I'm currently based, but the work that we do has a global mandate or global perspective, and we do cover most of these areas that you see on the map. So it's good to have this conversation with you, and knowing that I'm also speaking to a global audience that is likely from one or several of those countries that are on the screen. And I'll now go straight to my talk. And before I go into depth of the conversation, I wanted us to first put into perspective the costs that biological innovations have on crop production worldwide, not only crop, but also livestock production, and how that informs the work that we do on biological control. If you see the sections that I've highlighted, these are quite significant amounts when you think about it. In 2017, for instance, the cost of invasions reached more than 1.2 trillion. That's quite massive if you think about it. And when we talk about annual cost of 26.8 billion, that's quite substantial when you think about it. And this is equivalent to about 10.46% of Africa's GDP if you look at this damage costs and the expenditure spent on management. And there have been some projections that have estimated that in 2017, the mean annual cost could reach up to 162 billion. These are quite huge figures. And of course, we do know, given that we don't have as much data, sometimes it is lacking, that this cost could be strongly underestimated, that the damage could be, an impact could be quite more significant than what we have already documented. And so we come to biocontrol, and I would like to just take us through what we mean by biocontrol or biological control so that we can understand the concept. It is the science that deals with regulating hosts. So you basically introduce natural enemies to regulate their hosts. And so these natural enemies can be plant pests, which include parasitoids and predators, but sometimes they can also be pathogens. So you see here, you're talking about a pathogen that infects an insect pest, and so through infection, it regulates the population of that pest. We can have either classical biological control or augmentative biocontrol. I'll focus this talk largely on classical biocontrol. And when we talk about classical biocontrol, this is basically when we introduce a pest with exotic origins to manage a pest to manage a species that has also been introduced through exotic origins. And so what you would find is that if a pest invaded an area from another area, then you would go to the original home of where that pest came from and introduce that natural enemy that was responsible for causing suppression or getting that pest in its original home, and introduce it in the new environment where the pest has become introduced. And that's what we refer to biological control. And so the species that we introduced for playing that role are what we call biological control agents. I will not go so much into augmentative control because this relates both to classical, but also it has an element of using already existing natural enemies and multiplying them and releasing them to control their hosts. But I'll focus largely on classical, where we're introducing an agent outside its original home to control a new invasion. And when you think about biocontrol, and I mean, as practitioners, we talk about biocontrol, there have always been two schools of thought of why we do this kind of work and what kind of rules we need to follow when we do biocontrol. There's one school of thought that says in many instances, we get a lot of natural enemies, and also, of course, as well as invasive species, but we do get once in a while natural enemies that get introduced in an area accidentally. No one seems to know how they get to come into a new area. And so we always argue that it will not be proper to regulate their introduction, because in any case, they do sometimes find their way into new areas somehow, for some reason or another, through either accidentally or deliberately. And so we do argue that sometimes, why do we regulate, yet we still find these species getting into these new areas? But of course, the argument is always that biocontrol is a different case. The typical biocontrol is a different case, because in such instances, the release is intentional, and it gives us an opportunity to be able to assess the risk before release. And of course, when you talk about biocontrol, we are almost sure, almost 90% sure that we're going to result into establishment, because we are pretty much reassociating a pest with its natural enemy. So to a large extent, we're not gambling and trying to make assumptions whether or not a biocontrol is going to establish. Most likely it will. And so that's why we now go through various protocols that guide us on how to introduce biological control agents. When you think about biocontrol, it is not a new practice. A lot of biocontrol releases have been done over many, many years, both in Africa, but also other regions of the world. If you can look at the slide there, in total, we have had close to 6,000 introductions of biological entrants over many, many years. And to a large extent, many have been established. Some were temporary, but a large number of them, of course, have been temporary. I mean, including temporary ones. And in terms of target pests, close to 600 pests have been targeted for biocontrol, as and when this report came out in 2016. To date, of course, this number has increased because we are targeting a few more invasive species and targeting by release of new natural enemies. And of course, when you look at the number of biocontrol agents that have been successful, we have close to 650 that have actually been successful. And so it just could show that biocontrol is a worthwhile pest control method where the potential of establishment and control of the biocontrol agent, the pest that you're targeted is quite high. Over the years, if you look at biocontrol, the practice of biocontrol from the 1870s, you could see that it has been peaking as the science of biological control has gained momentum, and there is a great understanding of the role that exotic natural enemies play a role in regulating exotic pests. It saw a gradual increase in the 1870s, all the way to up to around the 1930s. And then there was a sharp decline. And that sharp decline you see around 1940, 1950 was largely due to the Second World War. Not much in terms of biological control and research was going on. So that could explain that decline. But after the World War II ended, then we did see a significant increase in terms of number of biological entrants that were being introduced to control a lot of invasive species that had invaded, of course, as well during the World War. Maybe one point I should make is that during times of conflict, there's a very high potential and risk of introduction of different species from one area to another. You have people moving around from one country to another. You have people who are moving from one region to another, carrying foodstuffs or carrying commodities that could easily bring in a new invasive. And so that explains why there was an increase in these invasions from the 1950s. But as we come towards the 2000s, you can see another decline in terms of biological control introductions. And this is where the catch now happens for us who work in the field of biological control, because this is around the time that the conventional biological diversity came into force, and a lot of the countries became signatory and started implementing the provisions of that convention. And this, in a way, made life difficult for us, because before it was much easier to introduce a biocontrol agent. You just needed to have partnerships with collaborators in the host country where you want to get the parasitoid, and of course be able to prove that this parasitoid can has efficacy on the hosts that you're trying to target it for, and sort of such a partnership introduced. But with the CBD, things have changed. You have to justify how the host country is going to benefit by giving you a biocontrol agent. You have to do more stringent tests to give you the kind of authorizations to do releases. And so work for us has been a bit difficult in terms of biocontrol, but nonetheless, it still goes on. And we are learning as we go along to be able to abide with some of these provisions of the CBD. And so what are some of the key features of biocontrol? Some things you should know is that biocontrol agents have little recoverable value. Whatever they can't be patented, like we do patent other commodities. So you can imagine bringing a natural enemy from Bolivia or any other country. This is a living organism. It's a species that you did not create as a scientist, and so you can't patent it. And so basically, when we do the work that we do, most of the work that we do is to introduce agents for social good, because they're not patented. Once they become established, they become self-sustaining. So they continue multiplying and breeding and reproducing, and over time sustain themselves on their own without any further additional release of new agents. Of course, like I've mentioned, yes, you have no products to sell, you can't patent. So once it has become established, it basically plays the role that it's supposed to play in terms of regulating the pest population. And of course, classical biocontrol creates a public good. So we release these biocontrol agents for public good. We are trying to solve problems that afflict our farmers, our smallholder farmers. And the benefits can be quite massive in terms of social, environmental. Many of the cases, biocontrol has resulted in reduction in pesticide use. And that is one of the key things why we seek to do the releases that we do of biocontrol agents for management of invasive crop pests. In terms of what could be some of the risks in classical biocontrol that we are also aware about, and do pay close attention to these potential risks one is significant reductions in the distribution or abundance of native non-target organisms. That's something we're always afraid of, that we could reduce abundance of native organisms. These biocontrol agents, through classical biocontrol, can also interfere in how effective these native natural enemies can be in terms of displacement. So if you introduce a natural enemy that is more effective or highly competitive than a native natural enemy, it could displace it and reduce on its efficacy. There are cases where you could have a natural enemy vectoring pathogens that may be harmful to native organisms. In fact, nowadays, before we release a parasitoid, when you apply for a permit to import a parasitoid, one of the things you are supposed to do is you're supposed to hold that parasitoid in quarantine when it arrives for up to 2 generations, just to make sure that that it's not carrying any pathogen that may be harmful to other native species. Other risks are intragrid predation or facultative hyperparasitism. This is also something that we as biocontrol specialists are worried about, introducing a parasitoid that may be carrying, and this is normally when you introduce using cocoons, you may find that the cocoons had been parasitized by another parasitoid, and so introducing other parasitoids that are harmful to the parasitoid you're trying to introduce is a risk that we always worry about. There could be effects on non-food requirements, such as effects on pollinators and seed dispersal of other species. We do worry about that. But of course, our main concern and biggest worry is extinction, when this classical biocontrol agent results into local extinction, local global extinction of other species. So this is our biggest worry. Most of the other risks that I've talked about sometimes could be reversible, but this one normally is irreversible, and it does cause us a lot of worry when we do practice releases and classical biocontrol programs. Classical biocontrol is mainly regulated by SPM3 of FAO. It regulates all releases worldwide, and it basically regulates, talks about export, shipment, import, release, and how these natural enemies are capable of self-replication. So it kind of provides guidelines on how you should do biological control, and provide the responsibility to IPPC and NPPOs to be as the responsible authorities for classical biocontrol programs. We do a number of assessments before release. For instance, we have to do constraint studies to determine whether the candidate biocontrol agent is a generalist or a specialist. Normally, we prefer that it is a specialist. And if it's a generalist, then maybe it is parasitizing other pests that are within the same genera, closely related. Or if they are similarly other pests, then that's normally not a point of concern. We also try to understand whether it will be established in the area of introduction. If it's likely not to be established, then there's no point in introducing. Issues to do with dispersal, issues to do with non-target effects, whether it's going to have any non-target effects, a point of concern for us, and also whether it's suitable to the environment in which you want to release it. And of course, we also ponder about redistribution and classical biocontrol. In some instances, you may find that you want to introduce a biocontrol agent, which is native, for instance, let's say to Africa. And for instance, I'm in Kenya, but the biocontrol agent is already in Uganda. And so sometimes drawing the line between classical biocontrol becomes thin, because even without any work on such an agent, it can easily move from Uganda to Kenya. And so it is just basically being redistributed. And so these are some of the things that we do risk assessment before we do the introductions. Of course, after talking about the risks, we also need to be aware about some of the benefits that classical biocontrol has had in the programs that we've run for many, many years. The typical ones that we'd like to give examples on is Cassava Mealybug, and give the example of Cassava Mealybug, which is a pest that we've worked on for many, many years. It was introduced in the 1970s, and we supported the release of Agaricus lopezi, which became established in 26 African countries. And of course, the benefits are there for everyone to see. Yields increased by 2.5 tons per hectare. And of course, the cost-benefit ratio was an investment of $1 to $149. So quite significant benefits from classical biocontrol program when you talk about Cassava mealybug. Mango mealybug is another classical example, which pest that got introduced into Africa in 1981 and was causing a lot of yield losses. In Benin, losses were estimated to be 89%. And in 1986, we did support a program for releasing a parasitoid in West Africa. And following the release, we did find that food production increased by 142%, with cost-benefit ratios of 1 to 145%. So quite significant benefits that we do find when we practice or implement classical biocontrol. In terms of water hyacinth, it was also a water weed that was first reported in the 1950s in the River Nile, and it did interfere with transportation on the rivers. And its cost at that time was estimated to be 84 million at its peak. And we did support the release of biocontrol agent, Ewivo, across Africa. And our data shows that following the release in a number of countries, the cost-benefit ratio was $1 investment, resulting in a benefit of 124 US dollar investment. So quite significant increments when you talk about the impacts and benefits of classical biocontrol. And of course, another example is the Papaya mealybug, something that we are currently doing in Kenya, but we've done in other regions elsewhere. This is a pest that causes up to 57% yield loss. And of course, the economic losses have been estimated to be close to $3,000 per hectare annually. And what's been happening is that farmers manage the pests using highly hazardous pesticides. And this was, of course, in Pakistan, CABI is still undertaking work on this pest, but the data that we have is largely from West Africa, where IITA did support the release of a parasitoid, Astravagus papaya, against this pest. And within a period of 3 years, it had reduced infestation by 83%. And saving producers close to 1.4 million US dollars, and consumers certainly similar amounts respectively. So you can certainly see that biocontrol does offer significant benefits where we've done these releases. In terms of stem borer and Riscasins that we've done for a number of pests, another example that I can give is for the spotted stalk borer in Africa. This is a pest that affects maize. And prior to the release of 2 parasitoids, Crustacea flavipes and Cassava stemata, we did conduct some studies on acceptability, on the performance. We also did do surveys on the parasitoids in the wild to see if there was going to be any non-target effects. And from those assessments, we did find that there are no effects on native stem borers, of course, which certainly showed that the fears that we may have had with regard to that introduction were certainly unfounded. And in terms of the benefits, those graphs do show the impact that this parasitoid has had in terms of maize production in the region. These are showing evidence from Kenya, Mozambique, and Zambia. So in terms of trends of crop production, crop populations under covered by the biocontrol agent, this has been increasing over time. And also in terms of poverty reduction across the 3 countries, you do see that the benefits have been quite significant. Prior to the releases in 1983, the poverty levels were quite low because farmers were suffering significant losses because of the stem borer. But over the years following the release, you do see that the poverty levels are going up because farmers can harvest more from their maize fields. And the same case has happened for mango production in terms of the Asian fruit fly, Crustacea dorsalis. If you see the graph in blue, this is around the time that the parasitoid was released, around 2010. And as you can see in red, the number of larvae in fruit. So we measure infestation in terms of number of larvae per fruit. You find that this has been declining. And so this again shows that the benefits can be quite dramatic, that within a short period of time, you can find significant benefits in terms of reduction of pest populations or fruit damage when you do release biocontrol agents for pest control. And this is another example of the diamondback moth. This is a pest that was exotic to Africa, and we did release a parasitoid from China, from Taiwan and China, for management of this particular pest. Diagnosis semiclosum was the parasitoid. And of course, following the release this parasitoid, these are some of the impacts that we have been able to document. There's been a reduction in terms of number of sprays of pesticides, in terms of the control costs, in terms of the yield, increasing to 4.7 tons per hectare. And of course, in many of the cases where we've done releases of these biocontrol agents, the cost-benefit ratio has always been a positive, that you do get a positive benefit when you do release biocontrol agents for pest regulation. And this is another case in Mozambique. Similarly, when you look at the light blue graph bars, this is the data in terms of farmers who are using pesticides before the parasitoid release for control of diamondback moth in cabbage. And you can see that at all the locations in Mozambique where this study was done, if you compare the baseline and the data that we had in 2015, in almost all cases, there was a reduction anywhere between 20 to 30, and in some instances up to 70% in reduction of pesticide use. If you can see an area like Boane, that is close to 75% reduction in pesticide use. So it does show that classical biocontrol and release of exotic agents do have significant benefits when it comes to pest regulation, but also controlling, resulting in better yield for the farmers. This is another case of the Liriomyza leaf miners, where parasitoids were introduced from Peru to control the leaf miner. And I just wanted to show this graph, especially the one at the bottom, because what you see in blue, in red rather, are the 3 exotic parasitoids that were released for control of the leaf miner. And what you see in black as the native natural enemies that were present during the survey, which were of course also causing suppression of the target pest. But the key thing to note is that many years after the release of the exotic agents, we can still find that even the native natural enemies are still existing. So the fears that the exotic natural enemies are going to displace or replace the native natural enemies has been unfounded. And so it just goes to show that sometimes the fears that we have prior to the release, when you do follow-up studies, you do demonstrate that this has not happened, that you still do find natural enemies still playing their role in natural pest regulation. And so as I'm nearly winding up, I would like to ask ourselves, and these are things that we always ask ourselves, whether we've had cases of serious risks happening with classical biocontrol elsewhere. We do have data that shows, and literature has shown that despite the many introductions of classical biocontrol agents for control of arthropods in many countries, and USA is one of the countries where this has been well studied, we found that less than 1% of these biocontrol agents have caused population level effects in non-targets. And only 3 to 5% may have caused some small effects, nothing significant. Yes, there are a few reports and a few publications that do suggest that may have been maybe 3 global extinctions linked to biocontrol agents, but these have not been proven. And when you take some time to understand how these extinctions may have occurred, in the 2 cases that I know about and have as much information as I can share, is that even for those species that are supposed to have been indicated as global extinctions, these were also species that had got introduced in an area and become naturalized over many years. So you can imagine if you get a new exotic species today, and 200 years down the road, it becomes extinct. Of course, over time, it has become naturalized and it becomes extinct. Then you ask yourself, was that really an extinction, or it's more of a case of an exotic agent that came and at some point got outcompeted and became extinct? So these are some of the things that we always ask ourselves about the risks that are out there when it comes to, of course, classical biocontrol. And so as I'm winding up, this should be my last slide, just to highlight for you some examples of work that we're doing on biocontrol. We still believe as CABI that biocontrol is one of the sustainable ways of managing invasive species. And so we're doing currently some work in Kenya on Asparagus papaya to manage the papaya mealybug. In Pakistan, we're trying to manage Parthenium using a parasitoid. We're doing the same for fallenworm in Kenya, Pakistan, and Benin using an exotic agent. In Kenya, we've done some work and are continuing biocontrol work on Opuntia and Gelmani using a parasitoid. And of course, the water lettuce in Kenya, we are trying to introduce a parasitoid called Rwomushana affinis. And maybe the last example is an interesting one as well that I would like to briefly highlight, because this is a biocontrol agent that was released in Kenya in the 1980s to control the water lettuce. But it would seem that that maybe it was very effective to an extent that water lettuce became almost managed in Kenya, and the parasitoid also disappeared is maybe the word I could use, or reduced to an extent that the population was not possible to be found if you did further surveys. And so we've had to do a new requests to apply for a permit for the regulator to re-import the parasitoid, because we've done surveys in the country and we cannot find the parasitoid anymore, yet we see that the water lettuce abundance is increasing. So this does show that certainly even when you do introduce biocontrol agents, in some instances it can be quite effective and result in an inability to find them in the future when you're trying to, of course, deal with the pest that you're trying to target. So with these remarks, I would like to come to the end of my presentation.
Excellent. Well, thank you so much for inviting me to talk today. This is a piece of work that we did between 2 and 3 years ago as part of John Mumford's environmental risk group in the Centre for Environmental Policy, working for target malaria. And whenever you do those 2-minute lift conversations and you talk about, the potential of gene drives and the potential of their effect on malaria, almost the first question that people ask is, what's it going to do to the other animals? So it's almost those very first questions are about ecosystem consequence. And these precise interventions are very interesting in the way that Ivan has been describing. We're not spreading nasty toxic things all over the place. We're actually focusing very carefully on one thing. So we have to think very carefully about the risk assessment associated with that one thing. So what does a precise intervention actually do? Well, it has the potential to change the abundance of that organism in the environment. And what will that do? So lots and lots of people have now heard about target malaria, and many people have posed these questions. And taking that on board has been part of the project's risk management plan from its very earliest days. So where do you start? Well, you start in the literature, because there's a very long study of Anopheles. There's been a lot in the 20th century. But even before then, people were writing about the malaria mosquito and thinking very carefully about what might happen and what might go on. But most of the studies that have been done are on the intraspecific ecology. So it's about the mosquito itself. There's a lot of studies on the biology, and a lot of the studies have focused on vector, so vector biology very specifically. But we also have some evidence that comes from monitoring and thinking about what's happened after vector control intervention, so more broader scale things. But the literature is not always easy. Over time, there has been some taxonomic evolution. The The complex of Anopheles mosquitoes has been more resolved, more species have been identified. So what Anopheles gambiae is called has changed slightly through time and become more precise. We now have molecular tools that enable us to work out exactly which species is which. But even in the modern literature, these are not always used. And Anopheles gambiae sensu lato, the broad complex, is still used in many circumstances. So there's a lot of evidence, but it's not always terribly clear. It doesn't always look at the community interactions, and there are taxonomic complexities over what you're actually looking at. So just a tiny general bit of ecology and broad ecology and consideration of what is an Anopheles mosquito as a resource to other animals in the community. So the density of these mosquitoes is usually highest around rural villages and sometimes on the outskirts of town and in small scale agricultural settings. Its density can be greatly enhanced by irrigation. So irrigated gardens and rice farming and rice paddies can raise the density of the mosquito compared to a more natural landscape really quite rapidly, because it provides an opportunity for larval development, enhanced larval development. But this is a very typical context for these mosquitoes. They're a little weed-like, they tend to be around human habitation. So what is the biomass? So one of the first questions we began to address, and this came in tandem with thinking about how many male mosquitoes are there, how many female mosquitoes are there, roughly 50-50. But beginning to look at what that presents as a resource, we did through calculating roughly how many there might be and multiplying that by how big they are, and find that in that part of West Africa where we were looking at the time around in Burkina Faso, that you're looking at a potential biomass of between 50 and 200 grams of mosquito per kilometre square in the wet season, and roughly a tenth of that in the dry season. So they're very seasonally respondent. But that is not much. It's not a great resource for anything else to actually depend upon. So that's thinking about the adult mosquitoes. But if you apply some very classical ecology with optimal foraging theory into there, we come across the basic principle that animals will invest in predating something if it returns more than they invest in getting it. So individual adult mosquitoes are very, very low return. So the optimal foraging theory predicts that no animal is really going to invest much effort in trying to catch an adult. They're a very low value and they're very disaggregated on the whole. There are two exceptions to that. There are male mosquito swarms where they come and aggregate and leck and whatever it is they do, show off in order to find females or for the females to find them. And recently blood-fed females. So recently blood-fed females tend to rest in houses, and they are easier to find and maybe more aggregated. So those are the two exceptions to that whole theory that they're a low value resource and they're very disaggregated, so it's not worth really investing much in finding them. Oops, sorry, I hit 2 there. But in the larval development habitats, they can be much more aggregated. So they tend to prefer, on the whole, not very developed water. They tend to prefer things like hoof prints and puddles where there isn't a great community. They tend to oviposit, and there is a preferential oviposition. So structured interactions, regular predation events are reduced because these mosquitoes lay their eggs in these small and ephemeral bodies, but they also avoid ovipositing in water containing predators, and which also have avoid high density of competitors too. They quite like low densities of competitors, but the higher densities they will avoid. So the female will go and look a bit further and find somewhere else to oviposit. Lots of the interspecific competition studies that we found in digging through the literature focus on other mosquitoes and the other mosquitoes that are present in these larval habitats. But a lot of the competition studies were performed in artificial cosms, and contrast them, so what you do in a jam jar in the lab, often contrasts with the results that you get in semi-field studies. And that limits the extrapolations we can make from conclusions drawn by entirely lab studies, because they sometimes do contrast quite substantially with the semi-field. There is now quite a lot of work going on and looking at what happens really for real in the field, but that is very much the next step, and here we're providing the underpinning effort. So what did we find? Well, we found that fish eat mosquitoes. So when mosquitoes do lay into areas where there's an established community, into rivers where there are fish, where there may be gambusia or tilapia, the fish certainly eat the larvae. But these are all insectivorous fish with a really diverse diet. And even those that have been proposed as biological control agents, and it does come up quite regularly that fish would be very good as biocontrol agents for mosquitoes. And in permanent water bodies, that may well be true. But the fish density doesn't vary really with the Anopheles larval density. Birds, people are very keen on thinking about birds. And certainly with the birds, waterfowl will eat them in the freshwater bodies. There's no doubt that waterfowl will opportunistically eat them. And as adults, many insectivorous birds will opportunistically take the mosquitoes. But the studies that have been done on diet, and there are some DNA type studies, and there are some where people have used seroprevalence studies, and all sorts of interesting studies in there. They found that in swallows and martins, they make up between 0 and 3% of the diet. And sand martins, they found evidence of the mosquito remnants in less than 1/3% of the samples they took from the sand martin. So they're not a popular diet item. They're an opportunistic diet item in many, many birds. Bats, people ask a lot about bats. Bats is one of the things that people focus on, that bats must eat mosquitoes. For the Anopheles mosquitoes, which tend to be around at dusk and then sit still for the night, there's a slight mismatch in opportunity. But also bats prefer the Lepidoptera. They like moths. They're much juicier, they're bigger, they give more bang for buck in terms of the reward there. Gonsalves and Bowman, they've done quite a bit of work on that. Gonsalves in Australia and Bowman in Swaziland. And the calculations that they make and the studies that they've done find that actually there's really relatively little mosquito in there. And where there is mosquito in samples, it's still a relatively small proportion of the diet. In big bats, it's almost negligible. But in some of the smaller bats, yes, they will have more mosquitoes in there. But they calculated that it would take about 600 mosquitoes to make the nutritional reward they would get from about 150 Lepidopteran flying moths. With the invertebrates, it's a whole different ballgame because things are much smaller and there's a lot of generalist predation going on there. The shoreflies, when we look at the other Diptera in the group, we've seen shoreflies that are observed predating adult mosquitoes, often females coming in to oviposit, but they're certainly very, very generalist. predators. And in the water, we get the Chiboridae, which are glassworms, and they're also opportunistic predators and will absolutely eat things like Daphnia too. So again, no findings of any specialism in that area. True bugs, same thing. Water boatmen will eat them. The guts contents analyses show that they're certainly consumed, but that there's no evidence at all of any specialism. Now, dragonflies do eat adults, and it's certainly a very spectacular sight watching an African mosquito swarm and the dragonflies, adult dragonflies, flying through. But again, they are very, very generalist. They may be the truest of generalist predators. Larvae, the same thing, opportunistic and voracious. They are voracious and sophisticated, but no evidence of specialism in the literature there. Shrimps, yeah, shrimps will eat them. And the flatworms will also eat them. There is interest in the spiders. So there are, for the larvae, there are some aquatic spiders which eat them, and there's peri-aquatic hunting spiders which walk around the edge and hunt on rafts of vegetation. But again, no evidence of specialization. So the one spider that comes up again and again, and Jackson has done quite a bit of work on this along with Nelson and Pollard, is this vampire spider, which has developed specialization on blood-fed mosquitoes. So many of you will know about the behavior of blood-fed mosquitoes, but when they fed, the females are extremely heavy and sluggish, and their wings don't carry them terribly far. So they tend to get to a nearby vertical surface and kind of hang on while they digest this meal. So not only are they really quite easy to catch at that point, but they're super rewarding because they're laden with blood. And this very cute salticid spider, it's a jumping spider, has developed some specialization on blood-fed mosquitoes. Now, they don't have to be an Anopheles. They will pretty much be any blood-fed mosquito that's there. And there's some evidence there that they make up, that the Anopheles mosquitoes make up a good proportion of the diet. But as I said, it doesn't have to be an Anopheles. It tends to be a blood-fed female mosquito, which is just so easy to catch and so juicy in reward. What about the other interactions? Well, there are many parasitic species, and I noticed that someone is going to talk about that in weeks to come, that it's highly likely that there are many parasitic species, and the most documented are the fungi and nematodes. There seems to be high infection rates in many of the natural habitats, but again, what I found at the time doesn't suggest any specialization on Anopheles gambiae. The interactions with competitor species is really interesting, but as I said early on, most of those interaction studies have been done in jam jars and petri dishes and in laboratory conditions. And those studies I'm reticent about translating those into grand conclusions in the field. There is some evidence also from the previous vector control situations where we see that other species may respond to reduction in another. And it's entirely possible that there is some competitive release in these larval habitats where reducing one species allows others to rise up in number. And there may be weaker competitive species that become more important if there was a successful reduction of the Anopheles gambiae. But on the whole, what we found across all this is that the Anopheles mosquito, this sort of low-value disaggregated resource that doesn't provide that much biomass per square kilometre, even in high-density areas, has no particularly strong and specific ecological links in the communities. I would really like to thank Jane Bonds, who was absolutely fabulous in digging through the literature. I'd like to thank Megan Quinlan and John Mumford, under whose hat this work came, and Target Malaria for funding the research and paying my salary and paying Jane while we did all this. I'd like to thank Mark Benedict for reading endless drafts and being very patient with me, and GeneConvene for inviting me to present this work. Thank you.
Thank you very much, Tilly, and thank you, Ivan, too, as well. And I'd like to open things up now for questions and answers, and we do have a few in the chat, and I think I'll go ahead and start with them. Ivan, this first one's for you, and it's the questions. I think I heard you correctly, but can you please clarify when you do risk assessment, your geographic scope is not just the country of initial import, but the potential range of, but is it the potential range of the introduced species? So can you just, and how do you predict the potential range?
Yeah, when we do the risk assessment looking at the geographical range, we are looking at the range in which this exotic parasite is going to establish. So we're looking at things like, let's say, like climate. We're looking at temperature. We're looking at the availability of suitable hosts. And so these are the kind of things we're looking at. And so what we normally do is, at that time when you're doing such risk assessment, of course, you've not yet done the introduction. And so you rely largely on modeling. So you will basically do some modeling and look at data in the country where this native, this parasitoid is native, and also compare that now with the country where you're doing the release. And, do some modeling, the ecological modeling using various tools, Maxent and others, and just try to understand if you did the release, what would be its potential distribution within the country. You find when you run these models, in some instances, the parasite will be able to establish in some parts of the country, but not others, because of maybe the temperature is too warm in others, or it's too cold in other parts of the country, or there are no host plants there that could be hosts for the pests, and subsequently not to be able to be available for parasitism by the parasite. So these are the kind of parameters we look at, but largely we do this through modeling.
Great, good, thanks. Tilly, I'm gonna throw one at you here. Raul Medina has a question, beginning with a comment. It says, not only may there be taxonomic complexities, but it's possible that genetic distinct populations within the same species may differ in traits relevant to pest control. Do you know if current mosquito control efforts are assessing how genetically distinct populations within the same mosquito species differ in traits that may affect their control?
Oh, that is a big question. The simple answer to that is no. I don't know. I think that there's quite a lot of work going on to try and understand the mosquito taxonomy and the genetic variation, and that you will have other people talking to you much more about mosquito genetics than I'm capable of doing. So I think I'm going to slightly bat that one on. I think, of course, it's important, but it's not my field.
Yeah, I wonder if I mean, it seems like perhaps the insecticide resistance and tracking the genetics of various insecticide resistance alleles across Africa might be an example of perhaps an example where intra-specific genetic variability is tracked that then impacts some control efforts. That might be one, just thinking off the top of my head. Okay, great. Thanks. Let's see, I'm going to go back up to Ivan here. Yes, here we go. So yeah, Nikki Faber has a comment here. So thank you for a very interesting talk. super interesting to hear about the biological success stories. And the question is, why do we only hear about the disasters with bullfrogs and others from Australia? Are people also, and here's a question, another one, are people also researching the usage of viruses for biological control?
Okay, yeah, just to go back to the first comment, I think we only get to hear about the bad things because that's what everyone wants to report. I mean, people are normally more interested in, I mean, that's why the media thrives, because they thrive on headlines and the bad news. And so that's why in many instances, we don't hear the good news and the good, I mean, the benefits that biocontrol can have, but there are certainly many if you do take time to read and understand over time and over many, many years how biocontrol has actually led into better pest management and pest suppression. Now, to go to the question, it is indeed true that we are also looking at viruses, and not just only viruses, but all kinds of pathogens, bacteria and fungi, and viruses for that matter for regulation of crop pests, and of course also dealing with invasive species for that matter. For instance, we are currently looking in Kenya. I mean, most of us do know that foraging is an invasive species that got into Africa from the Americas, and there's been lots of efforts trying to control these pests. And one of the things we're looking at is use of baculoviruses for management of these pests. There are a number of strains of Spodoptera polynucleohydrosis virus, SPMNVs, that have been found to be effective against falciparum. And so a number of companies, for example, have commercialized some of the strains. I do know two products. There's one by a company called Agbiotech that has patented and commercial producing a virus called Foraging that can be used on farm to control falciparum. And I think Andermatt, another company called Andermatt, has worked on one called Spodovir that can also be used for falciparum. So it is true that we're also looking at viruses in terms of how they can best be used to manage invasive pests. But the two products that I've talked about are of course, commercial products, but also when it comes to native or viruses that we already find existing in the environment. We've done some surveys where we've been able to find some viruses, baculoviruses for falciparum that do cause some mortality to the pests, though they don't have that high efficacy compared to the commercial products. And for the ones that we find locally existing, we do work with smallholder farmers and train them on how they can harvest the cadavers and produce their own viruses that they can continue spraying to deal with the pests in their field. So that's an area that we are currently working on, and it's going to be certainly, in addition to biocontrol and classical biocontrol, is also the future when it comes to biological control of crop pests.
Great, thanks. Tilly, here's a question: Would the density of mosquitoes increase in the rice field because of nitrogen-based fertilizers are applied, which in this case would enhance algal growth, the food source for the larvae? And is this something you might be able to throw some light on?
I think my instinct is that it would, and it would help not only just by increasing the food source, but it provides a more complex environment in which larvae can hide from predators. And there's some evidence that there are behavioral responses to predation, that different size larvae can get away differentially. But if the environment is more complex, then anything that happens to be wanting to eat them will have a harder time. So yes, the density of mosquitoes could potentially increase, both through increased nutrition and through a more complex habitat, yes.
Okay, great. Great. Ivan, here's a question for you. Is there a comparison between the effect on non-target species between biological control versus insecticide. So I guess have there been studies that look at non-target effects, the relative impacts of non-target species between those two control methods?
Yeah, that's a very interesting question for which some studies have done, but in terms of solid data, it's not very apparent because we do know that a lot of pesticides are used for broad spectrum pesticides for that matter, and that's pretty much why we end up going the biocontrol way, because you find that farmers are using lots of broad spectrum insecticides, some of them really nasty, some of them organophosphates that are banned, that don't not only affect the target pests, but also affect other non-target organisms. We did a study, for instance, in Kenya that found that on a crop like tomato, which is affected by the tomato leaf miner, you find that farmers are spraying up to 17, 18 times per season against tomato leaf miner. So spraying is no longer based on scouting and damage, or economic thresholds, but based on a calendar. So you find that farmers are having kind of where they spray per week against this pest. And they're using broad spectrum pesticides, which not only affect the target pest, but also non-target species, including those native species that we think that we would like to be protecting. So, and another classic example is a piece of work that we have started doing in northern Kenya. If you are aware, we've been having instances of desert locusts since 2020, and it's only lately that, of course, the desert locust is being managed, but it's being managed by spraying pesticides, largely ULVs, ultra low volume pesticides. And if you want to think about what ultra low volume pesticides means, it is a pesticide that has not been mixed with any solution. So a typical emulsifiable concentrate pesticide is mixed with water to dilute it to a certain concentration and spray. But when you talk of ULV, you're okay spraying a pesticide that is 100% concentration. And so we've been having aircrafts spraying against the desert locust in northern parts of Kenya. And what is certainly happening is that these pesticides are broad spectrum, and they're not only killing the locusts, but also killing other non-target species. By common sense, that is expected. And so what we're trying now to do is to do some kind of impact assessment and try to understand how has the biodiversity, changed as a result of these spray operations have been undertaken for desert locusts. So we do know that use of broad spectrum pesticides does have some effect on non-targets. We just need to get that evidence. I'm not saying it is there yet, but we are working on trying to make a case that certainly it is having biocontrol as a more sustainable measure for pest control is much better than use of pesticides, even when it comes to these native species that we are trying to protect.
Great. Thanks a lot. Tilly, John Mumford asks a question. He says, why do you think bats, birds, et cetera, stand out so much in the minds of the public as great predators of these mosquitoes, despite such a lack of evidence?
Yes, I think that it may have something to do with nature documentaries. I think it may have something to do with how we perceive bigger things as more vulnerable, which they may well be. And I think it's just what people know, that people's first instinct isn't, ooh, the water boatman. It's, ooh, the bats. And they're slightly romantic, and they're big, and they're known. So people associate them. I think I don't know, it must be about 18 months now, maybe slightly more since the paper came out. And it's a well-cited paper. People have approached me, and one person has said that we missed the lizards and that we should have had geckos in there. I think we didn't find anything really on geckos, so they're not in there. But I think I would look harder in retrospect. But what's really noteworthy is how little has come back to us saying, you missed this and you missed that. So I think that given that this was a pretty big literature survey and covered what we knew, we did pretty well to find lots and lots of this stuff. But you're right that undoubtedly people latch on to bigger things. They may latch on to something that they once saw eat a mosquito, and that little bit of anecdote is enough to drive a big belief in them.
Yeah, I guess in some cases people don't necessarily think quantitatively; they're thinking qualitatively, and so they see a connection and aren't necessarily thinking about the relative importance of particular connections. They just see that connection, and the connection's there, and therefore it's important.
Yes, I noticed there's a point about gambusia being made by Dr. Ghosh in
Yes.
the chat.
Yes.
And yes, I think gambusia are certainly effective at reducing mosquito larval density where they are coinciding. And I think if you have big permanent water bodies, they're probably very good. But in Africa, these Anopheles mosquitoes, in this context, tend to avoid those if they possibly can and go for the ephemeral and poorly populated water, which wouldn't support gambusia.
Yeah. Stephanie James asked a question here. Are there any useful parallels in how potential non-target effects are examined for biological control agents and how these issues might be addressed for GM mosquitoes? Maybe just to add a little to that too would be how are non-target effects assessed in a biological control before a biological control agent is decided to be deployed?
So normally what we do is you do some risk assessments. Even when you release, you want to import a biological control agent, there are a number of requirements that you're supposed to fulfill to the regulator before you're allowed to do the releases, of course, within the country. And so we have a checklist of the kind of tests that you have to do. First of all, and I did mention one of them. The first one is to prove that the biocontrol agent has not, by importing the biocontrol agent, you've not imported a hyperparasitoid or a pathogen, another pathogen, and so you'd have to basically rear this biocontrol agent in quarantine for at least two generations, just to make sure that you aren't carrying any pathogen or hyperparasitoid. And then you do a number of tests. These tests, first of all, include doing the efficacy test to see what is the parasitism rate of the parasitoid. If it is not very efficient, then there's no basis for doing release. So you must aim to have a level of parasitism 50% and above for you to make a justification that this is going to contribute to pest suppression. And then you also would need to do some host specificity tests. So here you're basically looking at testing this parasitoid on other species that are very closely related to this pest. So normally in the same genera. So maybe you're talking about noctuids, for instance, or tephridids. So you would select, identify a number of species that are within the same genera and do those tests to verify if or not this parasite is going to have those effects on those other species. Normally, if the effects extend to other species in the genera that are also pests, normally the regulator will not have an issue. Of course, they'll ask you to maybe expand the scope because it's now apparent that this natural enemy can parasitize more species within the genus. But if it's basically a generalist, and if it's a specialist and has a very restricted host range, then normally we will not have issues to prove that this parasitoid is going to have the negative impacts that we're talking about. And then, of course, you would, after you've done, you basically provide that information and done the test in the quarantine. Before you do the release, normally the regulator requires you to do releases in a small area. This is an area that would be normally isolated or quite distant from other parts of the country, because they would want to see if this pest is going to now how it behaves in a typical field situation when it's exposed to the the vagaries of nature, the environment, to temperature, rainfall, whether it's going to establish and whether the rates of parasitism you're getting in that area are or comparable to what you get in the lab. So these are a few examples of the kind of tests we do to prove or to give justification to the regulator allow us to release. And they are very strict. So you really have to provide evidence to get a basis for release. And so we do ensure that we are following all the requirements and ticking all the boxes and following the checklist before we can even ask, because even for CABI as an organization, we have a reputational risk to actually preserve and prevent and defend. And so we would never deliberately release another agent which we can prove, for instance, that it has significant non-target effects.
Yeah, great. Thank you. Could you say something a little bit more about how the decision-making process goes for releasing a biological agent? And I'm just thinking in terms of community engagement, which we hear a lot about with deploying technologies, and also issues of cross-boundary movement, which clearly, a biological control agent could cross boundaries. And so how are decisions made? Okay,
before we do releases of the biocontrol agent, in addition to, ticking the boxes in terms of the checklist, we really do have to consider talking about your transboundary movements. A lot of these biocontrol agents can move on their own. You see, once you release a biocontrol agent, you basically have no control of where it's going to move. Some of them are fast movers, so they move within a very short period of time to other areas, but others don't move as fast. And so, assuming that you're having an agent that has the capacity to cross boundaries, so it's a fast mover, then normally you would have to notify the neighboring countries that you're planning to do a release in your country. Of course, they would normally not have an objection, but you would have to basically do the releases. Notify them, and then you'd also prepare a dossier that you'd present to the regulatory agency. Normally, when that dossier is being reviewed, it's not reviewed by one agency. There are a number of agencies that review the dossier, from agriculture to environment to health, human health, to the fisheries people, so there are quite a number of broad stakeholders that you interface with, who review that dossier before they can give you the authorization to release. And when you talk about community engagement, it's good you should bring that up, because I do recall that when we did release Phopias arisanas against the Asian fruit fly, we didn't do, I do agree that at that time we didn't do very good community engagement, and the community saw us bringing wasps within a cage and releasing them in the field. Of course, we released at a farmer's field. And then a couple of months after that, there was an outbreak of malaria, and we got accused of having released mosquitoes into the environment carrying a strange malaria pathogen, something like that. That's like a classical case of what happens if you don't do a very good community engagement. And so what we do normally before we do any releases is to engage with the counties where we are going to release, do some programs on radio and television, do some leaflets, and hold some focus group discussions and have meetings with farmers where we do, engage with them and tell them about the parasitoid and what you are releasing, so they're also aware about what we're releasing. And so, a typical case of that Phopias arisanas, if we had known then, would have even had the community, putting their hands inside the cage and seeing that it's not going to, the mosquitoes, what they thought was a mosquito is not going to try to bite them or something like that. And so we are aware about this, and we do deliberately plan to engage with communities and the areas where we are going to release, that farmers are aware about what we're releasing and the potential benefits that these releases are going to have in their communities.
Great, thanks. I'm going to share a comment that John Munford shared with us. He says there's an interesting case in the UK at the moment where an exotic agent for control of a chestnut pest has been given trial release approval by the regulator, even though the exotic parasite has already entered the country, naturally, if you will, from abroad, and appears to have established in small areas. So the intentional extension of the establishment has needed regulatory approval. So
yeah, and this reminds me also of the case for Phopia sarisanas in the case of Mozambique and South Africa, because we did release Phopia sarisanas in Mozambique for many years before South Africa could allow to provide an import or a permit to do releases. But of course, Phopia sarisanas moves on its own, and some of the areas where it was released were parts of northern Mozambique, which border the Stellenbosch area, where you find citrus production. And so by the time that the South African regulator NAPOSA was giving approval for release, the parasite had pretty much crossed the border and was already into South Africa. So that's another example of where you can be deliberating whether to release or not, or give a permit to release, but already the parasite has crossed the borders and already reached your territory.
Yeah, very interesting. We have not too many other questions here, but I did want to ask you another one from my observations of your talk, listening to your talk. The timeline that you showed of the the use of biological control agents in that time series that you showed was quite interesting, and the precipitous decline in the use of classical biological control leading up to today was interesting. And you mentioned the Cartagena Protocol as being a factor, perhaps in accounting for part of that decline, but that decline seemed to have initiated a bit earlier than that, right? It seemed like it was already starting to go down in the 80s and the 90s in terms of the use. And I just was wondering if there were other factors within the insect control community, or that might have also contributed to that the decline in the popularity of that control method.
Yeah, well, I would like to think that even before Cartagena and the CBD, were adapted. Many years before that, the conversations had started on how do countries benefit when they provide natural enemies obtained from their country and introduced into another country. And I do recall around that time there were a number of cases where a lot of countries were patenting all kinds of things. You go to rainforest in the Amazon and you collect something and you move with it to another country and you patent it, yet it doesn't belong to you. So those conversations had started around many years before these conventions were actually assented to. And so a lot of countries certainly became a bit worried about exchanging their material without being exactly sure whether this is going to be patented and how it's going to benefit them, as a country. And of course, it is only later that, of course, when it comes to classical biocontrol, like I did mention in my talk, a lot of biocontrol agents cannot be patented, but there are a lot of products that could easily be patented. You could go to the Amazon and collect a certain plant or extract a certain oil or a certain something there, and you patent. So this conversation has started happening, and I think countries were becoming a bit worried about exchange of materials without being very clear about how they'll benefit. That would be my explanation why that decline may have as well happened.
Yeah, great.